Hello everybody, welcome to this video lecture on lichens. It is mainly for the benefit of English medium students. I am D. Rajashekar, lecturer in Botany, SGK Government Degree College, Vinikonda, Guntur district. Lichens are not a single organism, they are a combination of an alga and a fungus. It is a symbiotic association between these two organisms. The study of lichens is called lichenology. Tulane first identified them and they were widely studied by another scientist, Dibari. They are commonly known as rock flowers because they grow on bare rocks where no other vegetation is seen. By secreting some strong acids, they weather rocks, and by forming soil, they allow the growth of other plants. Hence, they are called rock flowers. And as they grow in the pollution free environment where no man activities are seen, that is why they are also called pollution indicators. When you find all lichen in a region, we can say that is free from all types of pollution. Hence, they are called pollution indicators. The algal part of lichens is called phycobiont. Phyco means algae, and the fungal part is called mycobiont. Myco means fungus. Generally, in the phycobiont region, either blue-green algal members or green algal members are seen. In the phycobiont part of lichens, either Ascomycotina fungal species or Basidiomycotina fungal species are present. Though lichens are the combination of an alga and a fungus, it is recognized as fungal organisms because major part of lichens thallus is occupied by the fungus and very little part by the algae. And also fungi carry reproduction and increase the population number, whereas algae, as they are rich in chlorophylls, they carry out photosynthesis and prepare food materials, and they are supplied to the growing fungi. This type of relation is called symbiotic association or mutualism, or also called helotism. Helotism means a master and a slave relation. In this relation, fungi is considered a master and the algae is considered a slave. Lichens are in different vibrant colors because of arsenic acid present in their body. They show worldwide distribution. So far, 500 genera and 13,500 species have been identified. Majority of them are terrestrial forms except a species, that is Peltigera. That is the only water lichen. Types of lichens. As per the habitat, there are different four types of lichens. They are saxicolous lichens, which grow on rocks, corticolous lichens, which grow on the barks of other trees, terricolous lichens, which grow in the soil, lignicolous lichens, which grow on the woods. As per the habit, there are again three types of lichens. They are crustose lichens, foliose lichens, and fruticose lichens. As you see in this slide, the first picture is crustose type of lichens, and second one is foliose, and the third one is fruticose. Crustose lichens, they are like a thin layer tightly adhere to the rock or substratum, so they seem like rocks. As they are tightly attached to the substratum, they cannot be easily disassociated from the substratum. They are like crust, hence the name crustose lichens. Example, Graphis and Lecanora. The second type is foliose lichens. They are little advanced than that of the crustose type. They are like leaves, hence the name foliose lichens. They show dorsiventral surface and their margins are like lobes. They are more or less like leaves, hence the name foliose lichens. They can be easily detached from the soil or the substratum where they grow because they do not tightly adhere with the substratum. Example, Paramelia and Peltigera. The third type of lichens are fruticose type of lichens. They are highly advanced type of lichens habit. They are like small shrubs. They may cause forest fires too when they get dry. They are with vertical branches or strap-like structures. They may go either vertically or hang down from the substratum. Cladonia and Afnia are the good examples for fruticose type of lichens. When you see the thallus anatomy in the lichens, we can see four different regions. They are, as you see in the slide, upper cortex, algal zone, medulla, and lower cortex. Upper cortex is with tightly coiled fungal hyphae and they are thick. Upper cortex is followed by a thin layer of algal zone. It is also known as gonadial layer, where we can see algal cells, followed by a wide region that is called medulla, where fungal hyphae are loosely coiled. It is followed by lower cortex, where again fungal hyphae are tightly coiled, but comparatively they are thinner than that of the upper cortex. From this lower cortex, rhizomes are developed. They are root-like structures. They closely attach to the substratum. Along with providing mechanical support to the plant body, they also provide nutrients. The lichen thallus anatomy, which is with all these variations, are generally known as heteromerous type of lichens. In some lichens, we cannot see the differentiation like this. In all dimensions, they are alike. Such lichens are called homomerous type of lichens. Lichens reproduce by vegetative method, asexual and sexual methods. Vegetative method of reproduction is done by either fragmentation or isidia or soridia or cephalodia. Asexual reproduction is carried by pycnidiospores, and asexual reproduction is highly advanced type of reproduction. In the ascolichens, it is just like the sexual reproduction of other ascomycotina fungi. In the basidiolichens, the sexual reproduction is more or less similar to that of the other basidiomycotina fungal species. The male sex organ is called spermogonium and the female sex organ is called ascogonium. Vegetative reproduction is carried by fragmentation. As lichens are fragile in nature, they can be easily broken down into small pieces by any physical means. Thus broken pieces of lichen thallus can be developed into new lichen thalli. Isidia, soridia and cephalodia are external outgrowths of lichen body. Structurally, they are little dissimilar, but functionally and anatomically they are all similar. Echidae are long club-shaped structures, and Soridia are powder-like or bud-like or plate-like structures, and the Cephalodia are wort-like structures. All of them have algal cells inside and enclosed by the fungal filaments. As they are all very delicate and fragile, they can be easily detached from the lichen thallus and get developed into new lichen thalli. Asexual reproduction is carried by Pycnidia. They are developed in the medulla region of lichen thallus. They are like flasks. These flask-like Pycnidia show long filaments in their on their inner surface. They are called pycnidiophores, asexual branches. They develop pycnidiospores at their apex. Pycnidiospores are haploid and uninucleated spores. They are formed in chains. When they are detached from the pycnidiophores, they come out freely from the apical pore of pycnidium on falling suitable substratum. When they get an algal partner, they can be developed into new lichen thalli. Sexual reproduction. In ascolichens, flask-like structures are developed in the medulla regions called spermagonium. They are like asexual pycnidium. These flask-shaped spermagonium develop spermatiophores on their inner surface, which in turn develop spermatia, male structures. These male spermatia come out through apical pore and attach to the ascogonium, female structures. The common wall where spermatia attached to the ascogonium dissolve so that the male nucleus of spermatia enters into the ascogonium. But karyogamy does not take place. This male nucleus and female nucleus undergo number of divisions and form many number of male and female nuclei. Meanwhile, ascogenous hyphae are developed. These ascogenous hyphae show apical ascus cells. The male and female nuclei in the ascus undergo fusion and form diploid nuclei. As these changes are happening around the sex organs, normal hyphae form a fruiting body that is called apothecium. It is a completely open fruiting body. It is like a cup. It shows three regions. The upper region is a fertile region where ascus and sterile filaments are present, followed by tightly coiled fungal hyphal region, that is subhymenium, which is followed by a wide region of fungal hyphae where these fungal hyphae are loosely coiled. This is called apothecium, a fruiting body. In the hymenium region, we can see number of asci. Each ascus shows diploid nuclei. It undergoes mitotic divisions and form 8 haploid nuclei. Each nucleus changes into an ascospore. Thus, each ascus contains 8 ascospores. When ascus breaks down, the ascospores come out, and when they get a suitable algal partner, they can be developed into new lichen thalli. In the bestiolike type, cellular fusion takes place. Male and female nuclei come closer, and later on they undergo fusion and form diploid nuclei. After unfavorable conditions, the cell wall ruptures so that the inner wall comes out like a tube where male and female nuclei fuse and form diploid nuclei. It undergoes meiotic divisions and form 4 nuclei. These nuclei are separated by septa. Each cell-like structure develops a steric on its peripheral region. Meanwhile, the nucleus in each cell undergoes mitotic divisions and form 2 haploid nuclei. One nucleus remains in the cell-like structure and the other nucleus enters into the sterigmata, which later changes into basidiospore. Thus, 4 basidiospores are developed. 2 are of plus strain and remaining 2 are of minus strain. When they get suitable algal partner, they can develop into new lichen thalli. Thank you for your patient listening. The important aspect and the remaining aspect of lichens is economic importance. That is discussed in the next video lecture. Till then, bye. Thank you.